Chapter 11, Section 2, Part 1, Applying Mendel's Principles. So nothing in life is certain. So if a parent has two different alleles for a certain gene, we can be sure that one of those alleles will be inherited by the offspring. However, we can't predict when it will happen, but we can figure out the odds. So Mendel carefully categorized and counted his offspring. And he used probability to explain the results of the crosses. So the definition of probability is the likelihood that a particular event will occur. So what are the chances that something will happen? That's what probability is. An example we can use is what is the chance of a coin being heads or tails? And we use coins because there's one or the other. And with Mendel, usually there was only two choices as far as the alleles go, either tall or short, either round or wrinkled, etc. So the chance of either outcome is going to be equal, one chance in two, so it's a 50-50 shot. Now, if you flip a coin 3 times in a row, what's the probability that it will land heads up every time? Well, each time is considered 1 out of 2, right? Because each one is an independent event. So if you flip it 3 times and it's a 1 out of 2 every time, the probability that you'll get heads 3 times in a row will be 1 out of 8. So remember that each time that you flip it is an independent event. That makes a difference in when we figure out probability. So just because you flip the 3 heads in a row doesn't mean that you're more likely to have a coin tail the next time. It's still a 1 out of 2 that 4th time. Now, obviously the likelihood that you have 4 in a row has gone down, but as far as having heads or tails, that has not changed. So when we say that alleles segregate during gamete formation, we're saying that they separate during gamete formation. You remember when we were talking about chromosome splitting in mitosis? Well, the same thing happens in meiosis, which is what gametes go through. Okay, so we can use the principles of probability to predict the outcomes. So in Mendel's plants, what was the result of the F1 cross? If you look at the picture, all of them, if you look at their phenotypes, they all came out. Oh, I'm sorry, the F1 cross, so we're looking at the F2, my bad. So they all came out either tall or short. So they either came out tall or short. And what were the gametes that were produced by the F1 plants? Well, one gamete would either have a big T, and the other gamete could have a little t. Now remember, it's only going to have half the number of alleles in it because we're talking about gametes. And what are the alleles for a short plant? It has to be little t, little t. And what is the chance of each F1 gamete carrying a T allele? That's right, it's 1 in 4. Okay, so there's 2 gametes, so the probability of both gametes having it is going to be 1/4. So 1/2 chance of having one little t from one plant, and then a 1/2 chance of having another little t from the other plant. So how many of the F2 offspring will be tall and how many will be short? That's right, 3 out of 4 or 1 out of 4. So the ratio for this would be a 3 to 1, 3 dominant to 1 recessive. And when in Mendel's experiments, this showed up constantly or consistently. So for each of his 7 crosses that he did, 3/4 of them showed the dominant allele. That's the trait that came out, was the one that goes with the dominant allele. And then about 1/4, you notice I said about, 1/4 of them showed the recessive allele. Now you know that not all organisms with the same characteristics will have the same combinations of alleles. Okay, because remember segregation, it happens randomly, so it won't always be exactly like that. So what alleles will produce a tall plant? Okay, big T, and big T, little t. And then what alleles produced a short plant? Little t, you know that. Organisms that have identical alleles, in other words, big T, or little t, are called homozygous. And if they are different, we have big T, which is called heterozygous. Now, as far as using probability to predict, the larger the number of offspring, the closer the results will be to the prediction. So, you might do 4 and not get a 3 to 1 ratio, but you might do 400 or 4,000 and get closer to that 3 to 1 ratio. Now, when we're describing their characteristics, we have their genetic makeup and then we have their physical trait. The phenotype is how something looks, or its physical traits. And the genotype is the genetic makeup, what the genes are, like big T, big T, little t. So what were the 3 genotypes of the plants, the possible genotypes? Big T, big T, little t, and little t, little t. So the genotype is inherited, and then the phenotype is formed because of the environment and the genotype. So what's that mean when I say because of the environment, the phenotype is formed? So you might have the gene to be heavy, for instance, say you have the gene to be overweight, but you're not overweight because you don't have enough food. Now, if you happen to eat enough food or eat too much food, then you may be more likely to gain weight, and vice versa. You might have, say, the skinny gene, but if you continue to overeat, it might override what that gene is. So 2 organisms could have the same phenotype, but different genotypes. So these 2 little dogs kind of look the same, but they still might have a different genotype. One could be, say, big T, and one big T, little t. And the way we can predict this is by using what's called a diagram called a Punnett square. It's named after a guy named Punnett, that's why the P is always capitalized, and it allows you to predict the genotype and phenotype mathematically. So when we make a Punnett square, you have to get the genotypes of the 2 organisms that will be the parents. So here we determine the allele. So one parent is a big B, the other parent is a big B, little b. So their possible alleles are going to be big B and little b for both of them. And then you draw a table with enough squares for each pair of gametes from each parent. Okay, so they got 4 possible gametes if you look at both parents, and we have our table. And this shows you how to fill the table for each individual or each possible genotype. Then, after you fill it in, you can see the genotypes. You also need to look at the phenotypes that were made. So in this case, the yellow ones would all be big B. In other words, they would have large beaks. So 3/4 of them would have large beaks, and 1/4 would be, 3/4 would be big beak, 1/4 would be small beak, and then one of them would be heterozygous dominant in the genotype, 50%, or 2 of them would be, did I say heterozygous? Heterozygous for big B, and then homozygous for little b, and then homozygous for B. I think I said that wrong. So it's a 1 to 2 to 1.